Alright, so be back, guys. In this video, we are going to talk about transcription in a bacterial cell. This is your girl Ruth, the .com scientist. Transcription. We basically have two occasions when DNA is copied or when there is any kind of replication. The first replication is when the entire genome is replicated. There you have DNA replicated, you get two exact copies of DNA. And this is basically done for purposes of species continuity. Before any cell divides, DNA has to be replicated such that the daughter cells have the exact DNA as their parent cells. The second replication, the second kind of copying which occurs is the transcription. And for the transcription, it's entirely meant to express the genes. It's meant to get results from the genes, to get proteins, basically. So here, DNA is copied to RNA, specifically messenger RNA, which is then translated into proteins. Gene expression, as I've said, it's basically meant to give us proteins. And these proteins are very essential for life sustenance of an individual cell. These are the proteins where these are the proteins, basically enzymes. Enzymes are needed very much for the sustainability of the animal. So all these proteins, we have the structural, we have enzymatic, we have so many. So all this comes as a result of the process called transcription, which is later translated to make the desired protein. Now, we have to note that in transcription, only a short segment of DNA is copied, because we should also keep it at the back of our minds that not all DNA is genes. DNA is also made of other, like some components of DNA are not even genes, not promoters. So basically, what we are trying to look at is to be able to copy that specific part of DNA which contains the information needed to make the protein of interest. Another difference between the transcription and the replication we've talked about is that in here the end product is ribonucleotides, basically RNA. The other one, the end product is DNA or deoxyribonucleotides. The steps in the replication, the very first one is initiation, and under initiation we find that we have the formation of the transcription complex. The enzyme which plays a key role in this is the RNA polymerase. RNA polymerase basically carries out the synthesis of RNA. And RNA polymerase, it's basically a multi-subunit enzyme, meaning it has many subunits in it. It has 2 alpha subunits, it has 1 beta subunit, and it has 1 beta prime subunit. The other subunit is called sigma, and all these subunits they assemble into one unit which is multimeric, meaning it has many subunits, and all of them together are called a holoenzyme. In different bacteria, you find that the sigma subunit varies, but the commonest one, or the major subunit that you look at in this is the 70, sigma 70. And it is this subunit, the sigma, which identifies the gene of interest. It is very important in this process. It recognizes the promoter sequences. We know that every gene has a promoter sequence just right next to it. Therefore, what the sigma subunit does is to recognize the promoter sequence, and immediately it is recognized the RNA that the holoenzyme is going to come and bind to that promoter sequence. Now, promoters are recognized by the sigma. As you have said, the sigma 70 is the commonest one. So promoters which are recognized by the sigma 70 usually have the TAT or the Pribno box sequence. It is TATAAT. So you need to get that. And it is approximately, so this is its location. It's approximately 10 base pairs upstream from the site of initiation of transcription, and the sequence, which is TTGACA, and it is 35 base pairs upstream. Now, what happens is that the moment the sigma 70, the sigma subunit recognizes the promoter, RNA polymerase is going to come and bind onto the promoter sequence, which is just close to the gene. The moment it binds, that specific part of DNA is going to start unwinding. It's going to open up, and when it opens up, the nucleotides which are complementary to the sense strand, or the strand that you are going to copy, they are going to start aligning just against the DNA strand. And the moment we form a few, the moment the sigma realizes that we have just our RNA, messenger RNA formation has started, it is going to dissociate. It's going to leave the rest of the units that we talked about, and it is going to leave 4 units. The moment the sigma subunit dissociates and goes away, we are going to find that we now have the core enzyme. The enzyme is now going to continue the transcription. This is what I basically talked about. We have the beta, we have the another beta prime, then we have 2 alpha subunits, and with the sigma subunit, we have our RNA polymerase is a holoenzyme. And the moment the sigma subunit dissociates, we remain with the core enzyme. Okay, so this is basically the structure of RNA polymerase. So now we are looking at the next step of the transcription, and which is, it's called the elongation step. Now we remember in the first step we just have a short segment of RNA which has been formed, but remember we need to get all the whole gene copied such that you can make DNA. We can make proteins using that specific information. Therefore, it has to get completed, and RNA polymerase will still bind to that part of DNA, and then the different nucleotides are going to come and bind until the messenger RNA is completed, and the binding takes place. The nucleotides are added on the 3' end of the RNA strand. You also have to note that. So now the last step in the transcription process is the termination step. Any process which starts needs to end at some point. And remember, we said that the gene is just a segment of DNA, meaning when that gene is completed in terms of formation of the messenger RNA, it has got to stop at some point. And we got to know that DNA has signals inbuilt to allow the RNA polymerase to identify the end of the gene it is transcribing. So this is very key. Now, how is the signal inbuilt within the DNA? In the DNA that we shall have an inverted repeat. an inverted repeat followed by a stretch of polyuridines, and it is a signal on the template that causes RNA polymerase to pause. It pauses. The moment RNA polymerase finds this inverted repeat, it's going to pause. If it was moving fast, it's first going to stop, and then after pausing, we have a specific protein which also comes in to play a role, and that is the Rho protein, which is abbreviated as P. The P is a helicase, and it binds to the C-rich regions in the RNA transcript. The RNA transcript, let's not forget, it is the messenger RNA that has been formed. The moment it binds to the RNA, it advances in the 5 to 3 prime. Remember, we've been adding nucleotides in the 3 to 5 prime direction, so this one is going to come from on opposite direction, and then it's going to start moving along our RNA transcript until it reaches the RNA polymerase. The moment it reaches the RNA polymerase, what happens next? We are going to see that the RNA polymerase is going to stall. Stalling is more like becomes extremely slow. It's wasting its time. That is basically the meaning of stall. And it stalls at the GC region, which is also the termination site. We need to note that GC, the hydrogen bondings between the GC are stronger because of the 3 hydrogen bonds which are formed between the guanine and cytosol. So the moment the RNA polymerase stall at this C-rich region, the P factor, it's going to unwind the transcript from the template, and then the messenger RNA is going to be released. So the moment the messenger RNA is released, that will mark the end of the transcription process. Thank you so much for listening. Please subscribe if you enjoyed this video and if you learned something. If you've got any questions, please leave them in the comment section. I will address them as soon as possible. Please like the video if you enjoyed and liked it, and then please click on the notification button if you want to see more of these videos. I'm going to put a link in the description that is going to have an animation, a bit of an animation, to help you get the real picture of what really happens, such that you can visualize it in your mind, and it's going to help a lot in remembering these facts. Thank you so much. See you in the next video of translation.